Many key diseases occur due to abnormal intracellular accumulation. Diseases like atherosclerosis, diseases like hemochromatosis, diseases like glycogen storage disorders or lysosomal storage disorders. All these diseases, they are basically due to some abnormal intracellular accumulation. Intracellular means within cell, some abnormal things accumulate. It could be lipid, it could be protein, it could be carbohydrate, sometimes it could be a pigment like hemosiderin or carbon or lipofuscin. So our basic objective in this InstaLecture number 117 is to learn that what are the key mechanisms. There are 4 key mechanisms actually, which leads to development of intercellular accumulation. So here we are going to understand the 4 key mechanisms which lead to intercellular accumulation, abnormal intercellular accumulation. So I already told you that many things could accumulate. Abnormal intercellular accumulation could occur. I would discuss this in a different InstaLecture, but I would give you a brief overview. In some diseases, there could be abnormal protein accumulation, intercellular accumulation. For example, tau protein intercellular accumulation could lead to neurofibrillary tangles in the Alzheimer's disease. Alpha-synuclein protein accumulation could lead to Lewy bodies in the Parkinson's disease. Even in the prion disease, abnormal form of intercellular protein accumulation occurs. Abnormal form of intracellular protein accumulation occurs in Russell bodies in cases of plasma cell neoplasms. So there are many diseases where the abnormal alpha-1 antitrypsin deficiency, another disease where the abnormal protein, intercellular protein accumulation could occur. Lipid accumulation, abnormal form of amount of triglyceride accumulation leads to fatty liver, steatosis. Abnormal accumulation, intercellular accumulation of the lipid inside the smooth muscle cells or in the macrophages, which are called foam cells, could lead to atherosclerosis. In glycogen storage diseases, we can get abnormal glycogen, form of glycogen accumulation. Some diseases, abnormal pigment accumulation could occur, like hemosiderin could accumulate in hemochromatosis, hereditary hemochromatosis, or acquired hemochromatosis in these conditions. So many abnormal intercellular accumulation, where there is an abnormal accumulation of protein, abnormal accumulation of lipid, abnormal accumulation of carbohydrate or pigment, could lead to many diseases. And here, now we are going to learn that what are the 4 key mechanisms through which this abnormal intercellular accumulation occurs. First cause of abnormal intercellular accumulation is enzyme deficiency. And here I'm going to explain this concept in the whiteboard. So let me go to the whiteboard first. So imagine that A, in an enzymatic reaction, biochemical enzymatic reaction, A is converted to B by an enzyme E. Now, if this E enzyme goes missing or deficient, what would occur? Which one would be increased inside the cell, A or B? Obviously, the answer would be A, because A cannot be converted anymore to B because there is a deficiency of this enzyme E. That is the first cause of abnormal intercellular accumulation. That means when there is some enzyme deficiency occurs in an inborn metabolic error. For example, lysosomal storage diseases, which you may have read in the biochemistry. A classic one is Gaucher disease. So in all these diseases, lysosomal storage disorders, the storyline consists of a deficiency of enzyme. And as this enzyme is deficient, what would happen that the precursor molecule, which is converted by the enzyme to another molecule, that molecule would be increased in accumulation inside the cell. So that is the concept of the enzyme deficiency which should be seen. And classic example which I mentioned is lysosomal storage disorders like Gaucher disease. Now, this is the first mechanism. So let me go back to the presentation again. So the first mechanism through which abnormal intracellular accumulation could occur is due to some enzyme deficiency. And as a result, abnormal form of substances could accumulate inside the cell. This could lead to cell death, tissue damage, and sometimes even to death also. This is the first mechanism number one. Mechanism number two is abnormal form of metabolism that could also lead to abnormal intracellular accumulation. A classic example is fatty liver or steatosis. Let me again go to the whiteboard to explain this. So, suppose this one is a hepatocyte. An abnormal form of triglyceride accumulation. If there is excessive triglyceride accumulation occurs in this hepatocyte, we call that condition steatosis, fatty liver. Question is that why fatty liver could occur? It's very simple. In the simple form, this could occur due to three reasons. Reason number one, if from peripheral fatty sources, fat sources, excess amount of free fatty acid comes there, that means excess free fatty acid comes into the liver, exported into the liver. Or second problem could occur if there is any deficiency or there is a disruption of the beta oxidation of the fatty acids inside the hepatocyte. That means these fats are not utilized properly inside the fatty acid oxidation that could lead to increased accumulation inside the fat cells. And the third problem could occur if there is any disruption in export of the fat from the liver cells to the outside, like if there is a disruption in the secretion of the VLDL from the hepatocytes. So either there is increased import or improper utilization or decreased or disruption in the export of the fat. And as a result, the fat starts to accumulate inside the liver cells in the form of triglyceride. This condition, this abnormal form of intracellular accumulation is called fatty liver steatosis. So this is an example of also intracellular accumulation. Another form of intracellular accumulation of the fat, particularly cholesterol, cholesterol could be atherosclerosis. Because atherosclerosis, what occurs that there is accumulation of this lipid occurring inside the smooth muscles and the macrophages. They're called foam cells, and when they're accumulating in the tunica intima of the aorta or other big arteries, that would lead to formation of the atherosclerotic plaque. That's also an example. Atherosclerosis is also an example of abnormal form of intracellular accumulation. Now let us go back to the presentation again to describe the other two key mechanisms. So already we explained two key mechanisms. One key mechanism of abnormal form of intracellular accumulation is enzyme deficiency, that is lysosomal storage disorder. Second is abnormal metabolism, which example is fatty liver. The third reason where there is an abnormal intracellular accumulation could occur when there is a defect in protein folding and transport. And this classic example is alpha-1 antitrypsin deficiency. Now, alpha-1 antitrypsin deficiency typically can present pathology in two organs. One is liver, another is lung. Why they occur? We try to understand. In the liver disease, the reason is this, that there is an abnormal folding and aggregation. This is the third mechanism of abnormal intracellular accumulation. If there is an abnormal folding and aggregation of some variant form of alpha-1 antitrypsin is seen, there is a mechanism called loop-seed polymerization that will lead to abnormal intrahepatocyte intracellular accumulation of alpha-1 antitrypsin. Typically, it occurs within the rough endoplasmic reticulum. And this form of abnormal folded aggregated variant of alpha-1 antitrypsin cannot be secreted from the liver cells. They get accumulated inside the hepatocyte, leading to the death of the hepatocytes. And that could lead to the liver pathology in the alpha antitrypsin deficiency, could lead to the development of even cirrhosis in the patients. And another problem which also occurs in the alpha antitrypsin deficiency, that is in the lung. Usually it leads to emphysema. How it leads to emphysema, let me explain this concept. So you know that alpha-1 antitrypsin deficiency, the name alpha-1 antitrypsin is basically quite confusing because it was named before its actual function was known. And actually in the lungs, it worked as an anti-elastase, anti-protease. So alpha-1 antitrypsin AT actually degrades elastase. And that's how it protects our lungs, the connective tissue in the lungs. And thus, and it protects connective tissue of the lung. What does elastase do? Elastase, as the name ends with H, you understand it's an enzyme, and it degrades elastin. And that is troublesome for us. That can cause a lot of problem for us because the lungs microarchitecture is basically made with elastin. So if there is a lot of elastase is there, that would destroy the lung and could lead to pathologies like emphysema. And alpha-1 antitrypsin protect us from elastase. It degrades elastase. So when it degrades elastase, obviously elastase cannot destroy elastin, and thus it protects the connective tissue of the lung and protect us from having diseases like emphysema, which is a classic chronic obstructive pulmonary disorder. But when there is a AAT deficiency, that could lead to development of early emphysema before 45 years of age. So what we learned that AAT abnormal intercellular accumulation of alpha-1 antitrypsin inside the liver cells could lead to two abnormalities. First, as they are abnormally accumulating inside the liver cells, it become toxic for the liver cells or hepatocytes, they would die and lead to liver pathologies like cirrhosis. Secondly, this condition, as alpha-1 antitrypsin is not secreted as they're abnormally accumulating, this misfolded form of alpha-1 antitrypsin is abnormally accumulating inside the hepatocytes. They are not secreted from the hepatocytes. They cannot go to the lungs, and that creates alpha-1 antitrypsin deficiency. What does alpha-1 antitrypsin do? It protects the connective tissue of the lung by destroying elastase. So when alpha-1 antitrypsin deficiency is occurring, they're not reaching the lungs where they're required. Elastase takes upper hand and destroy elastin, the microarchitectural framework of the lungs, and could lead to diseases like emphysema, which is a classic chronic obstructive pulmonary disease. And so they're causing problems both in the lung as well as liver. That is due to the abnormal intracellular accumulation of the alpha antitrypsin. So now let us go back to the presentation again. So now we learned that third mechanism through which abnormal intracellular accumulation could lead to diseases like in alpha antitrypsin deficiency. There is a defect in protein folding and transport. So there's a defect in protein folding and transport that could lead to abnormal form of proteins, like abnormal form of alpha antitrypsin gets accumulated inside the liver cell hepatocytes. The hepatocytes die due to this toxic accumulation, and also alpha antitrypsin is not secreted from those liver cells. So in the lungs, elastase takes upper hand, degrades elastin. Elastin makes the framework of the lungs. So it's a key, and that degrades the connective tissue of the lung, leading to formation of the emphysema disease. This is a classic example of abnormal intercellular accumulation leading to diseases. And this is the third mechanism where intercellular accumulation is occurring due to defect in protein folding and transport. And in alpha antitrypsin deficiency, intrahepatocyte accumulation occurs within the rough endoplasmic reticulum. And that's why you can see some in the liver biopsy, you see characteristic PAS positive diastrase resistant globules in the liver biopsy, which is a characteristic liver biopsy finding in the alpha antitrypsin deficiency. Fourth mechanism through which intercellular accumulation could occur is ingestion of indigestible materials. The idea is simple. If there is cell inside the cell, there is accumulation of some substances like carbon or silica. The cell has no mechanism, no enzyme to degrade it, to process it. So they would start to accumulate inside the cell. And sometimes it could lead to pathologies. For example, if there is silica accumulation inside the cell, that could lead to diseases like silicosis. It could elicit abnormal fibrosis. So this mechanism is purely due to ingestion of indigestible material. To digest that material, the cell has no machinery, no enzymatic machinery. The cell cannot handle it. So they start to accumulate there. So altogether, what we learned that there are 4 key mechanisms through which abnormal form of intercellular accumulation can occur. What are those? So let me summarize again. What are those 4 forms of abnormal intercellular accumulation? One is due to enzyme deficiency. Classic example, lysosomal storage disorders. Enzyme, as I give you example, A is converted to B by an enzyme, E. If E is missing, A would start to accumulate inside the cell. If it is abnormally accumulated, that will lead to the cell death, tissue injury, and sometimes even to death in certain diseases. These are typically seen in inborn metabolic errors. Some classic examples are lysosomal storage disorders. In the second mechanism, I told that there is an abnormality in the metabolism. So example I gave, fatty liver, steatosis, and how it develops. Third mechanism through which this abnormal intracellular accumulation could occur is when there are abnormally folded proteins. They accumulate, start inside the cell, like alpha antitrypsin accumulates inside the hepatocytes. And they are not secreted, so they are deficient in the lung where they are required, leading to emphysema, and the liver cells usually die due to apoptosis. And the fourth mechanism, which I told you, is when the cell, some abnormal form of substance like carbon or silica accumulating inside the cell, and the cell does not know, cell is clueless how to handle this because cell has no machinery to handle this. And these are the 4 key forms of the mechanisms through which abnormal intercellular accumulation occurs. Now, regarding the diseases which are associated with abnormal intercellular accumulation, I will discuss this in a different InstaLecture session. Thank you very much.